So while we are waiting, I can tell you what the plan afterwards is. So we want to head towards waypoint 5, which is
It's pretty
southeast, I think.
yeah, southeast of us.
Us, yeah.
Quite a distance.
Can you tell me how far away it is approximately?
Yes. Just give me
Yeah,
a
excellent.
minute.
Thank
Find
you.
it. Approximately, right now, it is 410 meters from us at a bearing of 155. And it's just about in the same depth contour.
I can put it on. Is it on NavG yet?
No, the next spot you're heading.
Yeah, that's nice to have
zoom way out. Is that?
Yeah, sure. We're sitting on the seabed, so I'm not using it. All better. Oh, that reminds me. What's that?
Are they chocolate chip? Yeah.
Oh, talking about snack time up here? Guys.
While you're waiting, Leela, folks want to know what about the sea stars is going to be studied. Is it for taxonomy or ecology or both?
Sure. Couple different things. We're going to look at there are different markers you can use in their tissues to try and figure out if they're consuming seep-derived carbon. So that's one thing. They're called stable isotopes that we look at. There's a specific signature of the carbon in organisms that are incorporating seep-derived carbon into their tissues. So Amanda Demopoulos at the USGS will be looking at that for us because she's awesome. And we also may look at their gut contents to see if they're consuming microbes that are present in the seep habitat. And we may also look at the fatty acids in their tissue for the same question. So just trying to look at the same question from a couple different angles. And then also actually the PMEL group that's doing the eDNA work will use those as some ground truthing. We'll actually
We are ready for this bottle.
Ready, ready?
Mm-hmm.
Here we go.
7158.
Triggered.
all right. Sample 25.
Thank you.
Yeah, and now on our way to waypoint 5, could we take another of these Niskins?
Oh, that's a fish.
Oh, yeah. Could we take another of these Niskins about 175 meters from here, so that we are like 200 meters away from the seep side when we trigger the last Niskin at the seafloor?
Yeah, can do.
Yeah, do you want to be on the seafloor when we do that? So should we just try to stop the vehicle and steady, make sure the water clears?
We can stay on the seafloor and just traverse, and I think it's like last time, if you can go 0.7-ish or something, that would be
0.7 knots.
Yeah, if that's possible with wind currents and everything.
Yeah. Does that sound good? If I make a one giant step, or do you want me to go 150 meters? I'll do the Niskin.
You can do one giant step. I could probably do it on the flat. Stop and pull kind of thing.
Bridge nav. Can you please step the ship 408 at 156 and a speed of 0.7 knots?
Oh, I meant the ROV, not the ship.
Gotta move them both.
Yeah.
The distance is 408 and the bearing is 156.
I'm gonna bring your head to 156, however you want to get there.
Are we good to turn the porch light back off?
Sure.
Bridge, you can go 0.5 knots on the ship. Sorry.
Oh, stuffed mud again?
Thank you.
Maybe it will wear off by the time we get there.
And you're tugging.
Yeah.
Okay, ship's moving.
Roger. Did I lose that scoop?
Did you lose the scoop and the pointer?
Looks like
Uh-oh.
it. Can't tell.
Should I stop the ship?
I see the pointer.
I think it was just a scoop.
Just a scoop. Who knows when that happened?
Over it.
Not getting it?
Oh, we'd look for it forever and never find it. Might get lucky, but unless you really need it for some sample.
Nope.
Tamara,
No, we are good, yeah.
I just want to confirm, you wanted it about halfway between where we were and waypoint.
Yeah, 200 meters away from where we took all the samples.
Okay.
Are you coming up?
Oh, I'm coming back down. Trying to, and what's going on here? You're good.
Dan, I'm going to make a new marker. That's where you're going to take the Niskin, okay?
Okay.
And then we'll replace that one after.
Coming back down, yeah?
Yeah, I'm trying to get some of that mud off, but it ain't budging.
You're also moving towards me.
Am I? Cannot be.
Okay, approximately there.
Oh, I see what I did.
It would actually be nice to be looking at the seafloor.
Yeah, sorry, I'm struggling here.
Okay, sorry.
Trying to clean the mud off again.
We still have some hydrate that is
Yeah.
coming off. So the Niskin bottle that we are going to take on the way is also for the eDNA to see what is happening when we get into the background and if we can still see the same signals that or still see the seep signals. And then afterwards, when we are at waypoint 5, this is an area where another bubble stream got detected last night during the multi-beam survey. So there's likely another seep site there with like not only bubble stream, but also the typical seep fauna. So we'll see what we are finding there. That's an area I think we have not been diving on in 2019.
Watching the bubble streams when we see them in the multi-beam is really exciting. Like, woo! But then when you get down here and you see how small they are.
Yeah, that's especially here with at depth when you have the hydrate skin. I think they pop up in the multi-beam way stronger than they actually are.
Yeah, it's kind of shocking.
They get bigger as they come up, do they?
It's a bit of both. The bubbles also get smaller because the methane is dissolving out, but in the same way they get bigger because there's less pressure. So it's a bit of an interplay. But they disappear at some point, or most of them. Only the shallow areas where the bubble reach the sea floor, the sea surface all the time. And with the deeper ones, sometimes we see them coming into the surface water.
What is that?
Is that a line?
It's another siphonophore.
Whoa, that's a
We
big
saw a
one.
couple of really big ones yesterday.
Cool.
No, are you sure?
Now we have a different kind of wrap.
Yeah.
Just hitching a ride.
How high in the water column do so many of the seeps tend to get? Because it seems like we can see them for a couple 100 meters in the multi-beam, but is that true?
Say again, couple 100 meters?
Yeah, how
Yeah,
high
that's
in the water
correct.
column? Yeah.
Yeah, some of them, yes. They have very high rise heights. They're different. Not all of them are the same. But yes, definitely. You can see them several 100 meters rising up, yeah. Have you been picking them, like during your shift at the multi-beam? Yeah, that's awesome.
Yeah, it's neat. They're like something else to look at.
Yeah. Yeah, and that's definitely always the interesting thing if you see something like remotely and then you can actually dive and have the camera at the seafloor and compare what you have seen like on the sensors.
Roger.
I know, just a little. Never mind.
Right, porch light on.
Yeah, it's crazy how the
Whatever you
combination of
took it.
instruments can take you from like the surface to the seafloor in less than 24 hours.
Yeah.
Actually, less than 12 hours.
It was
in this
very,
case.
yes,
Oh,
it was
I'm
very
behind
short.
you. Let's see what's happening. Try and get out in front here.
I was also coming about to take out a 6-head wrap.
Roger.
Like, better follow you.
About how far from the sampling location are we now, Annie?
We're approximately 80 meters,
Cool. Just keeping track in the log.
92 meters. Wait, we should clarify. You mean like how far from where we took sample 25 or how far from where we're going to take?
How far from where we took like the cluster of samples Not 25, though,
We're
like,
111 meters from there.
okay, cool, thanks.
Audio check.
What the?
Audio check.
Have some folks asking about the red blobs that we see floating. Those are jellyfish. I know at least some of them were Paralia jellies, and there are a lot of them. On cue, 3.
Good job.
And a lot of folks are asking what type of sea stars the red ones are. We are not entirely sure. Yeah, still trying to figure that out ourselves.
The jellyfish kind of look like flying saucers, like flying through
They
a
do.
starry
There were a
night.
couple earlier that were really like UFOs.
What, did you just turn us into turbo speed?
Yeah, I'm just trying to get everything lined up here a little.
You're approaching your Niskin site?
What's that?
You're approaching the Niskin site?
Yeah. We can take this one on the flight, can we, tomorrow?
Yeah, that's okay.
2 meter altitude, okay?
Yes, I think that's fine. Oh, actually, can you do 1 meter? Then they can compare better. It's better 1 meter.
Yeah, can do. Gonna give us the porch light. Thank you.
This will be Niskin 5.
It's pretty much there, is it?
Yep.
Triggered.
What sample number?
That is 26.
Coming up at the end of your leash.
Right there.
10, 50. approximately there, Dan.
What's that? Oh, yeah.
Next
Thank
waypoint.
you.
Amanda was giving some more fun facts about the acarax that might have been deeper down in the sediment at that last site where we saw the bubble streams. She was noting that actually the coating, so they have this coating on the outside of their shell. It's usually like brownish. That lasts for a while after the clam itself dies, and you can actually analyze that coating as well to deduce things about the nutritional, or where they were deriving their nutrition from, and also about the environment that they were living in. And you can age the carbonate that's inside the shell itself, and when you compare that to maybe a carbonate rock that you collected from right nearby, you can draw some conclusions about how long that seep has maybe been around, and how long those organisms have been using it as habitat. So maybe shells themselves are actually still useful to collect.
You tugging?
Really?
Yeah.
Thanks, Amanda, for throwing in all the fun facts to discuss.
When we get to waypoint 5, what are we? I think you may have said this. We're looking for the seep that we saw last night.
Yes,
And
exactly.
then
Yeah.
macrofauna.
Yeah. The first thing we will be looking out for the bubble streams again and see if there are bacterial mats, if we find some clams there. Oh, and also like compare how has it been looking at the waypoint we just have been, or at the two areas we have been, and if it's the same type or the same morphology, or if it's completely different ones. And then also we'll keep our eye out again for hydrate. So there's still a chance, we are still in the hydrate stability zone, so it's still possible that we find the solid phase of the methane at the seafloor exposed, so we can take one of those samples.
Nice. Are we going to take the cluster 2 of the push cores here as well?
Yes, that's the plan, yeah. So we hope to find, like, again, like we have taken the ones now in bacterial mats, so possibly if there is a different type of fauna, we will take them in a clam bed or with gastropods or something, or also we're totally happy to go for bacterial mats again, if that's what the typical prevailing type is that we have here from the ecosystem.
Cool, thank you. The ship's about 144 meters from position.
And we are also going to take our last Niskin over the seep.
Wrong button. You can stop the ship like 40 meters short, and we'll let Argus swing in so we don't run over. Stop the ship 40 meters short of the ROV target.
Yep. Nav. Bridge nav. Can you please change the step size to 75 meters at the same heading, 157? Thank you. What are you doing?
Knocking the mud off. Trying to break something.
Yeah. I'm gonna, when you're done with that, I'm gonna steal bubble.
Roger.
And you're tugging.
Really? Always pulling on Argus.
Annie, do you happen to know if anywhere in the Grafana type data there's lat long of either the ship or the vehicles?
In the Grafana, I do not know that, but I can give you that information.
I just have a scientist ashore asking if that's read out anywhere, and I don't know of it anywhere, but I feel like it would be somewhere.
That's super secret.
Yeah, I don't know.
Right, I
think
guess
that
you're
would be a
right.
Tim
We don't
question.
usually.
Okay, you can take it. We do have there's the overall map on one of the Grafana pages. You can kind of get a relationship of where we are in relation to Oregon.
Right.
I hate that map.
Oh. What happens when you tilt up just a bit on that one?
Oh, what?
The bubble camera. Yeah. Just enough. Yeah, perfect. I'm less likely to notice plowing.
Tugging.
Barely.
Ship's going to stop in 15 meters.
How many?
15, and then we'll be 40 meters from the site.
Roger.
And was waypoint 5 another spot where we had seen bubble streams in the water column data?
Yes, that's correct.
Oh,
Yeah,
cool.
we have seen already in previous years.
Mhm.
These have been seen, and also last night in the multi-beam survey. Yeah.
And they were huge signatures, which means that they could be teeny bubble streams or they could be big bubble streams.
Yeah, who knows? Yes. We're hoping for big one,
Yeah,
of course.
of course.
The weather is looking
Putting out all the big bubble stream vibes.
cooperative.
What's that, man?
I said the weather is looking cooperative.
There it is.
Yeah, we're at 18.5 knots right now.
Depends on which anemometer you look at.
I'm looking at the ship's anemometer.
Manometer?
They're all on the ship.
Touche. I'm looking at the bridge's anemometer.
I think
Manometer.
they're
Bridge
all on the
nav, you
bridge.
can hold the position right there. Thank you. Okay, ship is in position.
Roger. So we'll move in a little slower this time, and you can see Argus is still
Trailing.
trailing. So as it swings in, then we'll look for the bubble streams on orbit.
Sounds good.
Manometer.
Yep, that's how you say it.
That's how you say it, right?
Yeah,
Manometer.
You've got it perfectly. And anemone.
And Biscotti.
Whoa.
What's happening there?
Oh, look at that crab. That looks like a really big crab. Hi, Mr. Crab. I think Tanner crab, maybe?
Whoa.
You think so?
Zoom
Looks
in,
about the same color.
zoom in a bit.
They
and
like
pattern.
to stick around seeps.
Yeah. Amanda's typing. Maybe she'll come through with the answer.
Maybe a bit more there.
This one isn't running a marathon
That's
like
good,
the one
good,
yesterday.
thank you.
Is he missing a claw?
Can't see.
Yes.
More than
Oh.
one.
Only has like 3 legs on the left side, maybe?
It's been
A
fighting.
claw is indeed missing, as well as a
A
leg.
claw and a leg.
Oh,
okay.
buddy.
Oh, the stories. 2 separate occasions.
Okay, seen enough of the crab?
Yep.
Off we go. Slowly.
Full wide.
Thank you.
Is that another crab?
Yeah,
There on the right?
Yeah.
What is that? Oh, that's a huge crab.
A
That's
couple
another one, yeah.
large, really large
Yeah.
ones.
I could slide over there, but it's the opposite way I want to go. Okay, you want to bring your head around to the south, Michael?
All right. Time to see if you can see any constellation in these stars. We can find our way
Yeah.
back.
There's so many, yeah.
It's always so interesting, like what the organism is. There's always a lot of
Yeah.
one thing. Sometimes it's ophiuroids, brittle stars. Sometimes it's these sea stars, you know. Other times it's a bunch of sea cucumbers. It always seems to be different.
What did I just do? There you go.
I don't know what you just did. That's better.
That was deliberate.
Gives me an idea where I am in relation to Argus. I'm going to come up a bit, if you guys don't mind, and see if we can
Hunt.
pick up a bubble stream.
Yeah, sure. Let's do that.
How far are we from the waypoint? Like 15?
Yeah, approximately.
20, 22, I guess. 24? Depends on where Annie puts her mouse when she does it.
Yeah.
Ooh, there it is.
There's something.
Looks like an ultrasound on that screen.
Yes.
Same concept.
Our bubble stream baby.
Bubble stream
Yeah.
baby.
On the
What's your
sonar
range
as
for that,
well.
Mike?
Yeah.
Bubble stream baby.
Okay, so we're seeing it. What's that? What's the range on that? Is that like 20 meters out or something?
15 meters out.
15 meters out.
Okay, I'll come back down. We should be able to find it from there. I'll make a happy trade where we can see seabed and mud coming off of Herc.
They're tugging.
Oh, looks that's probably the depression there. You want to step the ship 20 meters south?
Bridge nav. Can you please step the ship 20 meters at 180? Thank you.
Oh, looks like a
Ship's moving.
little
Yeah,
Pac-Man
it's
thingy.
maybe one of those depressions. See how far Argus swung aft?
Mm-hmm.
It was way up there, and it.
And we're not actually that deep.
No, that pendulum effect gets worse as we go deeper. Oh, look, you can see the depression and the bubble stream on orbit.
Oh, yes.
I totally missed this depression. Is there a depression ahead of us more?
It looks like it. Yeah.
Oh, okay.
Looks, you can see
Oh,
it
yeah, I
there
see
on
that.
Mm-hmm.
Oh, that's cool.
What are those things called?
Which things?
So we're doing this a little slower this time. We wait for Argus to swing in and become plumb under the back of the ship and then do a small step in. Oh, I should have done it the first time. It's been a while.
Keep the current in our favor?
Probably not. But if we're not screaming in it, driving down the dirt road at 80 miles an hour, you don't have 2 miles of dust plume behind you. It follows you for a long time and displacing all that water in it. Stays suspended in the water column, and then if the current's not favorable, then it comes with you. But
I'm coming.
oh, getting impatient. Ship has moved 9 meters, so I should be able to move.
0 meters.
Got some slack in that tether there.
You should put thrusters on, like at least help it along a little.
Needs a tether out button. Ooh, so what is he at? Tinafor? Is that right?
Yep, there you go, Dan.
I think he's got his
Tinafor.
LEDs on.
Yeah, so those are they're also known as comb jellies commonly, and what you see glittering are the combs. So there are a couple of rows of combs along the side of the Tinafor, and that's what looks like glitter as the light catches it as those combs move.
Are they combing for food in the water column?
Yeah, I think so. Now I'm forgetting that, but I believe so.
Now the mats are coming up, bacterial
Oh,
mats.
yeah.
Yeah. Getting
so-
to the
Combing for
edge.
What's
a
that?
disco party. They're combing for a disco party. That's why they had the lights.
Well, there's a bunch
No.
of them.
Oh, wow.
And you should see bubbles in about 7 meters.
Just
Yeah.
kidding. So correction on the combs, that's for their propulsion. So they're using that to move.
Oh, neat. Thank you, onshore scientist. Or Leela, did you look up the
That may have been me Googling to try and remember.
Well, they actually,
Nothing wrong with that.
they're actually predators, right? And they get their victim in, and
What was that? What?
they're actually predators, aren't they?
I guess everything's kind of a predator. Yeah, it's eating something. But they're not like those the cubozoid, the jellies that like stalk their prey.
Right.
This is a 5 meter range. We should see bubbles.
Yeah.
Is this the depression maybe?
This
Yeah.
is the
Yeah.
depression.
Just damp
Wow.
right up to the side, to the right. Or, I'm sorry, to the starboard.
What's that weird stick thing?
Yeah.
Huh? That's the pokey prop.
Some ROV lost its pokey.
Maybe you should dig around for the scoop a little too.
I lost a scoop here a couple of years ago. Mach 1, I think. You want to go down in there or just fly over to the bubbles? That looks like they're on the other side.
Oh, there are bubbles right in front of us.
Fly.
Oh yeah, they're right there.
They're coming out of that depression.
They are. Of course they are. Ship is stopped. Argus is stopped. Okay, I'll see if I can get down in there.
Yeah, let's
you don't have a
look at
lot
them.
of leash.
Do you want me to move the ship a little?
I'd say 10.
Sure.
South?
Yeah.
Bridge nav? Can you please move 10 meters at 180? Okay, ship's moving.
Copy.
What
Is
is
it like
that
an old
stick?
marker?
Yeah, someone's been here before. Looks like an old
No,
pokey stick.
just a piece of rock.
It's an obelisk.
Of
Wood.
What does
wood,
it mean?
yeah.
Piece of wood.
It's like those ones they found out in Arizona a couple months ago.
Yeah.
I don't see any hydrate, so.
Mm. Can we zoom in the bubbles and also, oops. Wait.
Yeah, I'm just getting hurt to
Yeah.
settle down there without.
I want to take a Niskin here above the bubble stream. So I don't know if you want to do that first while we are still flying, or.
I'm in the mud now.
Oh, okay. Yeah, that's
You
fine
can,
then.
go ahead and zoom in, Nova.
Yeah,
Copy
that's
that.
perfect.
Hold on, I'll center it up. Okay.
What's on the yellow thing on the
I
top
don't
right?
know, yeah.
I've lost depth perception. It's gone.
Oh, poor fish just blinded. Now he's stunned.
Can we tilt the camera a little bit to
Yeah, you wanna
see
see where they're
where the
coming in.
bubbles come from? Yeah. If we can see.
Oh.
Oh, there, yeah.
Yeah.
Zoom in more there, Nova.
Sure thing.
Another tiny curtain.
It's like a dollhouse curtain.
Yeah.
Oop, oop, oop. And there's like a little bit of ones down here.
Yeah, yeah.
It is wild that that shows up so clearly in water column sonar data.
I think that's because they have a hydrate coating around them.
Oh, the hydrate
Yeah,
coating around the
yeah.
bubbles. Okay, all right.
Mm-hmm.
That makes sense.
And it could also be that there's more. But I mean, it's quite an intense one. I mean, yeah.
So is a hydrate coating almost like a tiny little chocolate shell, like as if these bubbles were M&Ms?
Technically.
Chocolate shell.
I like these analogies. This is good.
Ooh, I wonder if
Are
we got
you thinking about the
shells
chocolate chip cookies downstairs too? Because I know I'm thinking about this.
29 minutes?
I wonder if they'll do those chocolate bowls on the ice cream that we
I
did
was
last
thinking
time.
the
Oh, it's
same
ice cream
thing.
sundae tomorrow.
Oh, it's
Today?
ice cream on
Yeah,
Sunday?
it
I didn't
is.
know that.
It
No,
is.
Oh, I forgot.
It's today.
Yeah, it's
Yeah,
today.
today is ice cream sundae.
Oh, I thought it was like tomorrow.
Anyway, though, look at that bacterial mat.
Oh my God, it looks like a chocolate chip cookie.
We can zoom out again that you have the bigger frame.
Yeah, I'm getting hungry.
Okay, zoom out for us.
Coming out.
Are those snails edible?
Maybe zoom in just a bit.
Coming in.
That's good.
Yeah. Yeah, so not that I'm trying to sample this, but this might be some hydrate that is just sitting on like a little coating of the sediment, basically. It has a bit of different color than the bacteria mat we are looking at, but this is not like, I would need
You
at
would need more
least
than that.
2 centimeter wide layer of hydrate to get it sampled with
Zoom in a bit
hydrate
there.
sample.
Tamara, do you mind circling what
I
the
can
potential
do that.
hydrate
I
is?
was thinking this might be,
Can zoom in a bit
Oh
more
yeah,
but
Alright,
just
coming
some,
in.
like sitting
Full
on
zoom.
that
it.
almost looks like, yeah,
there. Is
It's,
that full zoom, is it?
it looks a little icier.
yeah. But maybe it's deceiving us again,
Yeah,
the mat. But
hard to tell. It looks so not matty. It doesn't look like there's the filamentous stuff flowing around or
Yeah.
anything.
I'm curious about this landing strip over here.
Yeah, that weird pole.
Yeah.
Pointing us in the direction.
Just like, what is this?
You need more to get with the hydrate sampler than that, don't you?
It wouldn't work here.
Oh,
Yeah, I would need a bigger, a wider layer of hydrate.
yeah.
I think this is,
Snowbank.
it
We
looks
need
more
a
like
snowbank.
a coating here, yeah, and
Does,
sediment
yeah.
beneath, yeah.
So is that sampler like almost like a pressurized scoop or core?
It's a two-piece thing. So one sample is like, I say a little drill, but it's like, it's not like electric or anything. It's just like mechanic. You place the drill like an ice drill in a way. You place it on the hydrate and then just turn it. It has little teeth at the end of the sampler, and then you turn the handle down and give a little just good.
Like compact it?
Yeah, not really pushing it in, but just sitting good on the hydrate, and then it drills into the hydrate and gives a small core that is about a little bit more than 1 centimeter long. And then once we have it in the sampler, we bring it back into the receiving module and click it into that, and then we are sealing it off with O-rings to make a gas tight seal.
That's so cool.
And then we bring it up as one. The receiver and the sampler as one piece, and can hook it up on our high vacuum extraction line that we have on board the ship here to get the gas out. So on the way up from the seafloor to the ship, the hydrate will dissociate into gas phase and the water phase, and that's what we can subsample then once we have it back on board.
Neat, thank
And
you.
then we also manage with using the high vacuum extraction line, we make sure that it doesn't get exposed to air at any time, and also that we don't lose any fraction of the gas. So we'll have like the complete sample that we can then subsample from, and even go for noble gases and all the total chemistry of
That's
that.
really
So
cool.
that's yeah. And the idea behind is to look at the fingerprinting method to make a difference between bubbles that are rising from dissociating hydrate and bubbles that are rising from free gas or free gas pockets, because we don't really know if the hydrate is actively dissociating or if it's free gas pockets that are generating these bubble streams.
That's really interesting.
Yeah, it's fascinating how much you can find out from one sample.
Yeah, we have started. So there are experiments from the lab already, but like this type of noble gas samples, they haven't been taken in the field yet. So we are trying to do that with this gas tight sampler. So when I say not yet, we did actually try it once 3 years ago here at the Cascadia Margin to test the sampler. Then it was brand new, and now it would be the second time that we are using this sampler. Yeah. So we don't have any scientific results yet. We are more working on the method still. And, yeah, developing.
Oh, very cool.
All this. Yeah.
So our plan here is
Niskin?
Yeah, so we're going to take a Niskin above the bubble stream, just 1 meter above the seep. That will be our last empty bottle, yes. And then sampling again for biology.
Okay.
There's is that a scale worm there? A little worm creeping along at the bottom of the
that could be
screen.
This guy?
Yeah.
Yeah.
Oh, good eyes.
It's been moving.
Yeah.
Slowly. Okay, are you
yeah, we
you
can
want to watch more? Or you
no, we can zoom out, that's okay.
Okay, no, you can zoom out. Yeah.
All the way?
Yeah, sure.
Full way?
Thank you.
Yeah, and now
Now the Niskin?
Yeah.
Okay.
And that would be the 1 meter above seafloor, just in the bubble stream.
Copy. Can you turn the porch light on for me? Thank you. Watch out for that milk crate.
You're going to go up a meter, right?
Correct. You make a note in the log that we're heavy ballast.
Say that again.
Make a note in the log that we're heavy. Or should float.
What is this? What dive? 5'9"?
Ballast. We need to
It's 1859.
look at the ballast a little closer. Sometimes our spreadsheet. Does a strange calculation. Or we miss something. Like the hydrate sampler.
Well, good thing you dropped the scoop or you'd be like way heavy.
Totally.
I know.
Actually, the scoop is buoyant, so.
Oh, maybe we'll find it at the surface.
Well, it had some bolts on it, so.
Make up your mind.
Well, one of the ones we first learned why it has the bolts on them, because if you drop them, they just float away.
But is it dropping if it floats away?
No, not really, I guess.
It's flying with style.
It's falling up.
Letting go.
Oh, that's a...
And it changed this over to auto depth. Maybe that will behave a little better.
How high are we trying to get?
Trying to get to 1 meter.
We're like 4 meters.
I know. Where'd they go?
We will go.
Are we cleaning out the area or cleaning out the...
No, I came up too high and then coming back down, dusted us, so I'm backing off a little and sneak up on it again and then.
They should just approximately be like right in front of us, the way you're looking right now.
Yep, it should be.
Now over to your right, maybe.
Unless Argus has moved.
I don't think so.
I don't think
Assuming
so.
that's a thing right there.
Yeah.
But I don't think he moved like 10 meters or nothing. Highly
Bouncing
unlikely.
around a little bit, but not much.
Highly unlikely.
Try again. I see what I did.
We find the pointy stick again.
It's right under the,
right under us.
yeah, right under the front left corner of the vehicle there. You look to the left of the bubble, you can probably see in the hole right now.
Mm-hmm.
There it is. There's a landing strip. What does it mean?
Aliens.
There they are. Well, they were in the Niskin.
Is there any
Nope.
chance?
You want the bubbles going through the Niskin, right?
Close to the Niskin. They don't need to go like through.
What is that?
It was what?
Are those the
There they are.
bubbles?
Okay, I'm gonna pull it. You happy there?
Yep.
Triggered.
Roger.
Thank you.
Sample
Okay.
number?
That was 27.
Thank you.
Oh man, look how dirty it is over there, right by the 5-arm.
Yeah.
I was just going to say, is there any chance that if gas were to get into the Niskin, that the expanding gas would unseat the Niskin stoppers at all?
It would,
They should
not.
tight enough to handle that.
and if they do, it'd be pushing water out. It'd be pushing the water out rather than
Right, then in.
than taking any in. Yeah.
All right. So we have the Niskin. What's next?
Okay. We could, ready for cores?
Yes.
Yeah. We'll collect 2 cores, similar over, the gray-white area adjacent to this, somewhere that's good to set down. And we'll take one of the ported cores, or the last ported core, and one of the regular ones.
Okay.
What are the ported core force?
Those are the ones that have the holes down the side so that we can extract pore water.
What?
Yeah, it's really cool. There's like these tiny little
holes that go
yeah.
at regular increments down the side of the core liner.
Yeah, I've seen that.
suck it
Yeah.
out.
And we can suck the water out from each of those layers and then analyze that water for the chemistry.
Got you. Got you.
Have you guys been live streaming the lab work